Dr. David Perlmutter. Welcome again to the Empowering Neurologist. You know, though it seems conjectural, there is no biological law that mandates that we must age. Aging is not necessarily inevitable. Now, that might raise eyebrows, but that's what leading science is really exploring at this time and really coming up with some incredible information as we uncover the basics of aging, what is going on, and how we might extend our lifespans, and more importantly, how we might extend our health spans. We're going to speak today with Dr. David Sinclair. He's the author of a new book, Lifespan: Why We Age and Why We Don't Have to. What a title that is. Let me tell you a little bit about Dr. David Sinclair. He is professor of genetics at Harvard Medical School, and he is the founding director of the Paul F. Glenn Center for the Biological Mechanisms of Aging, again at Harvard. He is clearly one of the leading innovators of his generation, listed by Time magazine as one of the 100 most influential people in the world in 2014, and then in 2018, again, listed by Time magazine as one of the top 50 most important influential people in the area specifically of healthcare. He is a board member of the American Federation for Aging Research. He is a founding editor of the Journal of Aging and has received more than 35 awards for his research on something called Resveratrol, which we will certainly discuss today, as well as something called NAD, something else that we are definitely going to be exploring today. His goal has been to look at reprogramming the whole aging process to actually reverse that process. And this has been hailed as truly a major scientific breakthrough. In 2018, he was awarded an Officer of the Order of Australia, which is basically the equivalent of being knighted, for his work on national security matters and human longevity. Dr. Sinclair and his work have been featured on programs like 60 Minutes, Today, The Wall Street Journal, The New York Times, Fortune, and as mentioned, Newsweek, among so many others. We are absolutely delighted to have Dr. David Sinclair join us today on The Empowering Neurologist. Dr. Sinclair, delighted to have you on the program today.
Thanks, David. It's great to be here.
So two Davids, here we go. I want to start off with really a point in your life, and it was 3 years into your time at MIT, when you were able to demonstrate increased longevity in yeast. So I'd like you, if you could walk us through that, and then let's have just a moment's discussion as to why on earth working with yeast would be important for all of us.
Yeah, well, so we're going back to the early 1990s now. And we didn't have the tools to manipulate human cells. We could barely change one gene. But in yeast, we could change a gene overnight. And so, kids like me in my early 20s, we learned genetics using baker's yeast. And on the side, we would make beer and meat in the warm room. So that was an added bonus. But yeast cells are eukaryotes. They have 16 chromosomes, or 32 if they mate and fuse. And they actually function very similar to our own cells. And that sounds crazy, right? Because this is a fungus versus a mammalian animal like us. But the basic building blocks of how life works are very similar. They have linear chromosomes, not like bacteria, which are circular. They get hungry. They need to find a mate. They starve to death. They have a lifespan. They age. They get old. They actually get big and fat and round and slow and sterile. And so I trained during my PhD years in Sydney, Australia, learning about a disease called maple syrup urine disease. And you can guess what the symptom is of that disease. And I cloned 3 genes in yeast cells that were identical, well, very closely matched to the human ones. And so I was of the mindset already that yeast could tell us a lot about human diseases. And I had a fascination with aging. And at the time, no one was doing in a rigorous way the genetics of aging and longevity. So I thought this was my opportunity.
So what we've learned then is, and I think in your book you made a very good point about it, that there's a lot of similarity, that 70% of the genes that yeast have are shared by humans, and that what you discovered in terms of this mechanism whereby that damage in the yeast cell causes this translocation of things that we're going to talk about away from a mating phenotype into a basically survival or repair the damage type of phenotype. And that has implications. You then studied this in rodents and worked your way through higher organisms. So where are we now in the application of that science that you discovered? How do we work through this translocation, basically the movement, if you will, of what were then identified sirtuin genes, sirtuin enzymes rather, coded by the genes? How do we get to the place of really getting our arms around the whole notion of sirtuins, which is, I think when you look in the book under sirtuins, we see your pictures. So you are the one to talk to about that.
Right. So this sterility of old yeast cells, it turns out that these sirtuins, these enzymes, normally they control a variety of cell functions, including mating. And during aging, they get distracted primarily by DNA damage. And they leave the place on the chromosome and go and help repair the DNA. But in doing so, and while they're distracted, the yeast cells are sterile. And that was the big finding in the 1990s. When I left MIT and started my own lab at Harvard, I wondered what could be similar about that process going on in our cells, and have spent literally the last 20 years trying to figure that out. And now we know that these sirtuin enzymes, there are 7 in our bodies, there are 5 in yeast, 7 in ours. They're called SIRT1 to 7. The SIR part of it's interesting. SIR stands for Silent Information Regulator number 2. And the information is important. We'll get back to that, of course. But what we now know is that these sirtuins can be activated naturally by being a little bit hungry or exercising. Caloric restriction is one way. But we also know you can activate them with chemicals. There are natural and synthetic ones that we've worked on. And also, because these are, at least 3 of them, control the way genes are turned on and off, we call this, gene expression. We think that the changes in gene expression as we get older, in part due to the distraction of sirtuins, among other things that occur, and it gets worse as we get older and with more DNA damage distracting them. That's, I believe, the major driver of aging, certainly one of the major drivers. And where we're at now are 2 things. In humans, we've got a clinical trial trying to activate the sirtuins that we have in our bodies already. And then we have what we call epigenetic reprogramming technology that we think is sending them back to where they came from. They're now permanently distracted in a very old mouse and probably a human. And we finally have some handle on how to get them to go back and reset the cell's age and get them to turn on the genes the way cells did when they were young.
So what you're saying is then there's a physical distraction or translocation of these sirtuin genes aware from, perhaps we could say, they belong to do good things, to going to other places to do other things that are basically repairing damage. For example, we know that double-stranded DNA breakage seems to be a powerful signal for sirtuin to move away from where it's, shall I say, needed or doing good things, distracting it, and this might be one of the hallmarks of aging, the fact that our sirtuins are dislocated.
Well, yeah, that's exactly right. And often, including myself, people ask the question, why would the cell be set up this way? It makes no sense. If they didn't do that, wouldn't we live for 1,000 years or more? And the best answer I can come up with that fits with the data is that it's a very ancient system, right? Remember, it's in yeast cells as well. So it's over 1 billion years old. And what I think it's doing is it's coordinating gene control with DNA repair. So when the sirtuins and other proteins that interact with them go to help repair, the genes that come on are temporarily beneficial. These are stress responses, DNA repair factors. In the case of yeast, becoming sterile is a good thing. You don't want to be mating while you have to repair your broken chromosome. But the problem, I think, is that though the system does reset, it doesn't fully reset 100%. And so one analogy is during Hurricane Katrina, the emergency response troops went down to to New Orleans. And then not all of them went back to their homes, and their homes became, full of weeds and didn't pay the bills. And if you keep doing that over, in the case of our lifetime, 80 years or more, then you'll end up with a huge mess in the country. Or in the case of the cell, a lot of genes that are now dysregulated, and the cells lose their ability to function and eventually lose their own identity and forget what type of cells they are.
We're going to get to that. I mean, that's I think that's probably leading edge of where we are. But let me take it back to the notion that the sirtuin relocates for one reason is to this area of damaged DNA. And there was a time when the accumulation of DNA damage was thought to be the main driver of the aging process. So in a sense, though, your newer understanding of the aging process does kind of envelope the whole notion of damaged DNA and other inputs in terms of sirtuin relocation.
Yeah, it does. This is what's been rewarding about the data that's come out over time. There's of course there's mutations that occur, and there's lots of DNA damage. I'm from Australia originally, and all you have to do is look at Australian skin, people who have sunbaked, or Floridians, as I mentioned. You can tell that it certainly accelerates the signs of aging, and you can accelerate what we call the DNA methylation clock that way. But unfortunately, antioxidants and DNA repair factors haven't been very successful at extending the lifespan of any species, even yeast cells. So there was some component of this idea that came out mostly in the 1950s and 60s when the world was obsessed with radiation and the effects of it, that's been unsatisfying. And what we've come up with is the proposal that DNA damage is important. And so getting exposed to UV light in the sun, of course, you want to avoid that for numerous reasons, including cancer and aging. But there's this other component that's actually the main result of that process, and that's the changes to the epigenome, the factors that control the gene expression, the cell's identity. And they're intimately tied together. They're both, so the genome gets damaged and the epigenome responds and doesn't fully reset. And over time, they end up causing cells to lose their function. Perhaps, David, it would be good if I explain a little bit about the epigenome.
Absolutely. So, we've heard our viewers are somewhat familiar with it, and maybe we go back to Waddington. Maybe we'll go back to the notion and explore your analog versus digital kind of the metaphor about, and look at the responsiveness, therefore, of our genetic code to our environment. That would be terrific.
Right. Well, actually, so the epigenome was very popular, again, in the 1950s. So Conrad Waddington proposed, in his efforts to explain how is it possible that a fertilized egg turns into an organism, a baby with 26 billion cells that know what they're supposed to do from essentially the same genome. How is that possible? So he came up with the cellular equivalent of a recipe. And what actually happens in physical reality is that the DNA is looped in big loops and spools, and it controls in each different cell type which genes should stay on and should stay off. And there are chemical marks that dictate that this is a gene that should stay on for 90 years in the brain or more to make that cell a neuron, for example, or a skin cell or a stem cell in say, the muscle. But what Waddington had no idea was that this was going on. That they were barely getting a grasp on DNA. They didn't know these other processes. So what he came up was a metaphor, which was that the fertilized cell is like a ball at the top of a mountain that would roll down and not randomly, but be directed into valleys where the ball would rest or the cell would rest. And that is a stable cell type. And if you're in a valley for a neuron, you cannot easily jump over the hill and become a skin cell. And that leads to the embryo and the baby. But he didn't know what the valleys and the hills were at the time. And now we know it's these proteins and DNA modifications like DNA methylation that create those hills and valleys that last almost the life of the individual. But as I mentioned, in the case of yeast through to humans, we see that those hills and valleys start to flatten out so that cells start to wander over into the wrong valley.
So then what leads to aging then is this sense that the methylations are in not exactly the right place, and that causes gene expression that doesn't bode well for us, leading to things like loss of proteostasis, mitochondrial dysfunction, exhaustion of stem cells, altered intercellular communication, all of the things that people seem to focus on these now as mechanisms of aging. But what you're saying, though, is this is the primary event. This is the upstream event, is this dislocation, this change in the methylation pattern of our epigenome that then it correlates so well with aging as, well, let's talk about the work of Dr. Stephen Horvath then. He looks at various patterns of how these methyl groups look in terms of their disbursement on the entire epigenome, and is able quite handily to correlate that with our rate of aging, our state of aging, pretty close correlation with chronological aging. But it's more or less a DNA methylation aging kind of clock that bears his eponymously is called the Horvath clock. And so that has, at least until recently, in Horvath's description, was pretty much indelible. I mean, we're kind of jumping the gun here on some of your newer research, but this whole idea that the changes in this methylation pattern really, may I say, represents our aging, and is really the upstream event that then filters down to all of these other mechanisms that researchers are exploring as leverage points to deal with aging.
Yeah, so it's still a theory, the information theory of aging, as I call it. It's emerging in the field, so we're right on the cutting edge, and over the next 5 to 10 years, we'll see where it settles out in the landscape. But what is making me more excited about this theory and its ability to explain a lot of these things are that, first of all, we know that we can clone an animal from an adult cell. Take a skin cell of a sheep or a dog or a cat. Barbara Streisand cloned her dog and got a couple more.
It wasn't in your book, I don't think.
Yeah, so that, I mean, if you've got enough money, you can do it. And what that really tells you, more than some people have a lot of money, is that the instructions to restart life, to go back to age 0, is in a cell. Maybe not every cell. Some cells may be damaged irreparably. There may be mitochondrial DNA that's lost. I'm not saying that the DNA doesn't have mutations, but the mere fact that you can clone an animal is remarkable. That means that you can reset the epigenome and start life again. So Stephen Horvath's work was remarkable too. He's a mathematician who became a biologist, and he was really not that into understanding how it works. He used machine learning to develop a clock based on which of these methyl groups correlated with the age of many hundreds, now thousands of humans. And he could draw a straight line, which is super interesting, but it doesn't tell you anything about why it changes over time and if it's important. And that's where our work and some of my colleagues comes in, where we were trying to understand what's driving these methylation changes. And as I mentioned, DNA damage is, we think, a major driver of that and the distraction of the sirtuins. And now we're at the point where we're asking the question, well, are these changes just a marker of aging, as though they're a hand on a clock where if you change the hand of the clock, push it backwards, it's not really going to change time? Or if Horvath's clock is relevant, if you push that hand back and make his clock younger, does the tissue, the animal, one day the human, become actually younger and healthier?
Well, I think that let's take a step here and talk about your recent paper that did demonstrate that using these Yamanaka factors, which were apparently designed to do that, to change the methylation pattern and revert the methylation pattern back to a less defined or more pluripotential scenario. Maybe walk us through first that original work. I think it was 2006 or 2004. And why did they do that work? And then let's go moving forward to how you were able to leverage that in terms of your research.
Right. So Yamanaka, Shinya Yamanaka, is now world famous for his experiment looking for transcription factors mainly that are turned on during embryogenesis. And he and his team looked at many different combinations and hit upon a combination of 4 factors, which are, of course, now known as Yamanaka factors. And they're called OCT4, SOX2, KLF4, and CMEC. CMEC is an important one to point out because it's also a potent oncogene, which is dangerous. And that's the main reason that Dr. Yamanaka confessed to me that he didn't think it was sensible to put his genes in an adult. But as you'll see, it actually might work. So what he did was ask the question, can you do the cloning experiment, but in the dish? Can you get an adult cell to be age 0 again? And if you can do that, you would have a pluripotent stem cell that could be turned into other tissues. So cloning in the dish. And it worked. He was able to create from many different cell types, including skin, these stem cells that we now use to generate little mini brains called cerebral organoids and new tissues, and inject these also back into patients. So he won the Nobel Prize with John Gordon, who did the tadpole nuclear cloning experiment. In 2016, they won the Nobel Prize. Now, a few labs took that finding from Yamanaka and asked the question, what would happen if you did something like that, not in the dish, but in a tissue or a cell from an animal and put it back into the animal? What would happen? And that was a big risk, right? Because you could easily end up with a cancer, a teratoma, basically cells growing like an embryo, but in the animal, which does happen actually if you turn on all of the Yamanaka factors. But we tried it anyway, and actually this paper that you referred to, David, was our breakthrough.
So what you did, it's hard to get my arms around it. You used a viral vector, wasn't it, to administer these factors into a rodent that you had traumatized the optic nerve. And then by administering OS and K, 3 of the 4 factors, you were able to revert these cells back to a less defined type of cell, basically induce them, can I say, to become somewhat like stem cells?
I wouldn't put it that way.
Okay, tell me.
So there are 2 states that we're finding. If you go all the way back to what Yamanaka did, then you're losing the cell's identity. There's an intermediate step that we're using where cells get to a younger state, but they stop.
Okay, so they do remember what they're supposed to do. I needed to get clarified on that. So they're not going all the way back. They're going back to their youth, let's say, where they already are. They know what they're going to be when they grow up, and now they get a second chance. And typically we say that these neurons, when they're damaged, that's it. Certainly, there's an implication here for the treatment of a spinal cord injury, brain injury, et cetera. But you used the eye because it seems to be at least somewhat immunologically segregated from the rest of the body. And it was an easy access point for you to actually demonstrate this regeneration from a youthful state of functioning neurons, in this case, in the optic nerve.
Yeah, that's right. We're interested in aging in every tissue. The eye was a good model system to start with. The virus, the AAV, the domesticated virus that is used in gene therapy, was pioneered in the eye. So we thought that was a good place to start. And also young nerves, very young nerves in neonates still regenerate, but young kids don't. So we thought, well, let's see if we could do that. But often I'm asked the question, and it's an important one, did we choose the eye because it had a better chance of working? And the answer is no. We could have tried to regenerate the liver, the skin, even the brain. And we only chose the eye, not only, but one of the reasons actually is that the student in the lab who did the work mainly, Yuan Cheng Lu, his family has an interest in the eye and has a company that is trying to use stem cells to reverse vision loss. And so he had that passion. I said, okay, go for it. I think the liver would be better. But I'm glad he did that because it's a very clear reading when you see the optic nerve grow back. But that was the first experiment. We went on to treat glaucoma and even old age vision loss.
So glaucoma, is something that's certainly very important. If this were translatable, is a pretty widespread situation in the elderly, a very important cause of loss of vision, along with macular degeneration. So that's the translatable goal here. Can we look at a figure from your paper, and maybe you can walk us through it?
Yeah, so this is the traumatized optic nerve that was pinched. And there's an orange dye that you can see that when the nerves are dying back unhealthy, those nerves don't take up the dye. And so the untreated eye will lose its vision, similar to the breaking of a spinal cord. What we did in the other mouse was to turn on the 3 Yamanaka genes, O, S, and K, for 3 weeks and have a look what happened. And remember, David, this is after the damage had occurred. We weren't prepping the cells. And what we found, and this was a remarkable day, my student texted me the result. You could see that the nerves were growing back towards the brain. In fact, if we leave them for 16 weeks, many of those nerves actually make it all the way back to where they meet the brain again, the chiasm, which is a first. This hasn't been done before. There's very little you can do for damaged nerves. But then we looked at the age of those cells, and 2 interesting things came out. One was that the damage accelerated aging according to the Horvath clock, which we didn't expect. I mean, who knew that damage would accelerate aging? But it does. And then the reprogramming factors delayed and reversed that age so that they became young and could grow back again.
When I was 19, that was was a while ago, I was involved in research looking at limb regeneration in newts. And a newt, when you remove one of its limbs, will develop what's called a blastema, which is this whole collection of these pluripotential cells that then ultimately know how to differentiate into the various tissues that are necessary to regrow that limb. And, who hasn't puzzled over that? Who hasn't wondered what is going on? And this research that you're doing is pretty darn exciting in terms of the potential moving forward. Let's move back a little bit, if we can, back to sirtuins, if we can, and the function of sirtuins and how important they are, the 7 sirtuins that have been characterized for within that influence the mitochondria, for influence the nucleus. But let's look at, if we can, the notion that we actually do have some control over the activity of the sirtuin pathways. And maybe we can talk about that in terms of how our lifestyle choices might affect sirtuins.
Right. So this is now going back to the yeast days. What we showed when I first joined Harvard was that if you make a hungry yeast cell, it will make more of a molecule called NAD. The long form of NAD, if you spell it out, is nicotinamide adenine dinucleotide. And it's a small chemical that cells use for 2 reasons. One is it's used for 450 or so chemical reactions. Without it, we're dead probably in 20, 30 seconds. But the other thing that cells use it for, yeast and humans, is to gauge cellular stress, adversity. And we found that when cells were hungry or they had too much salt or too few amino acids, they made more of this NAD. And the sirtuins, they don't work unless they have NAD. They have their own enzymatic reaction that absolutely requires NAD to work. It's called deacetylation, and that's how they control chromatin and the epigenome. And so as levels in the yeast cells rose in response to being hungry, the sirtuins in the cell came on, more active, preserved the epigenome, improved DNA repair, and the cells lived longer by about 30%. And we had papers in the early 2000s showing that something very similar happens in mammals as well, in rats at least, and now in humans we know that when you're hungry, you exercise, levels of NAD go up, and we hypothesize that they also protect the body. A great example of that in mice was an experiment done by my good friend and colleague Shin Imai from Washington University in St. Louis. His team made a mouse where he turned on the sirtuin gene number 1, SIRT1, in a mouse's neurons, and that mouse lived longer, which is great news. And so what I think is going on is when our body perceives adversity, a million years ago, our ancestors were often hungry and had to walk or run a lot. This turns on the survival program that keeps the epigenome in a younger state, repairs DNA, and does other healthy things as well.
Well, NAD was a really boring molecule in the day. I mean, when we learned about NAD back in the day, it was involved in energy. That was maybe we spent a day on it. But now, as is so often the case, we're seeing that molecules have a lot of different roles in the body, despite their original characterization or even how they're named. So what you're saying then is that the activity of sirtuins is NAD dependent, and we really depend on having good sirtuin activities to do the various things that you've now described. And I think you mentioned that we can increase NAD levels, which decline with age and are seen to be declining in people who have chronic degenerative conditions. Interestingly, that we'll get to this perhaps, that seem to predispose them to bad outcome with COVID. But nonetheless, because of this decline in NAD, it might be in our interest to do whatever we can to bump our NAD levels. So how do we go about that? What can our viewers think about to do to increase their NAD levels?
So what I do is I try to skip 1 or 2 meals a day. And I wouldn't say this for very young people, certainly not teenagers, but I'm 51 now. I don't need 3 full meals a day and snacks and dessert. So I'm eating maybe 1.5 meals, focusing on plants. I try not to eat too much meat, though I do eat meat. Don't eat too much sugar. Sugar spikes will suppress your sirtuin activity, we think. The other thing you can do is simulate exercise. Well, lift weights is what I do. I do a little bit of running, try to lose my breath. Just 10, 15 minutes twice a week is enough, actually, we think, to turn on these defenses. You can do something artificial, which is what I do. I take a precursor to NAD, which forces the body, we know from human studies, to make more of this NAD molecule. The one that I take is called NMN. There's another one called NR, which are available as supplements. And in clinical trials, we've seen that can raise NAD levels about 2-fold. If you're a marathon runner, by the way, you probably only have NAD levels about 50% higher. So, you could argue that I'm higher than a marathon runner. I certainly couldn't, David, run a marathon, but I do think, based on an N of 1, as we say, I can run further than you would otherwise think based on my level of training. So that's one thing. I still take my good old molecule Resveratrol. Anyone who's followed my career knows Resveratrol came out in the early 2000s from my lab as an activator of SIRT1. We then showed that you could give Resveratrol to old and fat mice, and it helped them. They lived longer, they were healthier. And we've gone all the way. It was controversial because the molecule is what we would say a dirty molecule. It was doing other things as well. But we're just writing up a manuscript now, David, where first of all, we had a science paper in 2013 that showed that resveratrol does activate the enzyme directly. This was controversial, but we showed it does. And you could mutate or change one amino acid in that enzyme, and it blocked resveratrol's activity. We've now made a mouse that lacks that one amino acid in the SIRT1 enzyme and given it resveratrol and a high-fat diet, and lo and behold, that mouse didn't respond to resveratrol. It couldn't run further. It wasn't healthy. So that's my, full stop period in that debate. Resveratrol does work, at least in large part, through resveratrol for lifespan. So I continue to take resveratrol every day, and those are my two main things, among the other things that I list in my book on page 304.
Well done. And as I said to you in our setup, that I went through 2 highlighter pens, and I'm not kidding you. It was there's a lot to learn. You correlate or kind of discuss resveratrol as getting this activity to happen, and then the NAD is sort of hitting the accelerator.
Yeah, that's the double combo. Yeah, I think of the NAD as the gas and the resveratrol as the accelerator, and together they work well. And part of the problem is that we think we lose NAD as we get older. If you take a skin sample, you have about 50% less at age 50 as you did when you were 20 on average. And we also think there's an endogenous, a natural activator that we make as we, in our bodies. We haven't found what it is. The closest that is the candidate is actually oleic acid. Doug Marcinek published a paper last year, remarkable, that oleic acid works exactly the same way as resveratrol. And when you change this amino acid, it's not only blocking resveratrol and the drugs that were in clinical trials, but also oleic acid's activity. So I've actually started taking oleic acid as well. Oleic acid, you might say, well, that's in olive oil. What's that got to do with anything? Well, first of all, it might help explain why olive oil and avocados are good for you. But it also turns out oleic acid is made in our bodies when we're hungry from the breakdown of white fat. And so if that story is true, it certainly would bring all of this knowledge together into one theory.
So we can take a precursor for NAD, nicotinamide mononucleotide, nicotinamide riboside. You take nicotinamide mononucleotide. We can exercise, that bumps up our NAD levels. And we can expose ourselves to heat and cold. There was a picture of you standing out in the cold. And we interviewed, I think you posted this past week, Mr. Wim Hof, who is the Iceman.
That was a great interview. Anyone who hasn't listened to it, you have to.
It was great. I immediately jumped in the pool, but I live in Florida, so I wasn't that cold. So anyway, but having said that, we know that the conversion then of nicotinamide mononucleotide into NAD plus there's an enzyme, nicotinamide phosphoribosyltransferase, transferase, that seems to be amped up by either the heat, being stressed by heat environment, or being stressed, low-grade stress, we call that hormesis in a positive way, of being in the cold. So what else can we do to bump NAD, aside from hope we don't get older?
Well, those are the main things. Metformin might do it. There's some overlap between AMPK, which is one of the targets of metformin, among other things. But those are the main things that I do. And the oleic acid may
okay,
help.
So and I'm going to say a significant number of people in the longevity world are taking a drug. And the reason I ask that is we had a show of hands. I was on the founding board of X Prize Longevity. And at our very first meeting, they asked how many of you are taking metformin, a drug. And surprisingly, I was very surprised by the number of really smart people who are taking metformin, adding David Sinclair to that list. And I'm tempted as well. But I will, maybe we can just explain why you're taking this AMP kinase activator, why you think it's looking at risk-benefit ratio. You've made a decision to take it.
Right. So unlike yourself, I'm just a PhD, so I don't make recommendations even for supplements, but certainly not drugs. But what I have been excited and convinced as a scientist about is the data. Right, I'm driven by data. I'm also, by the way, influenced by smart colleagues and friends. And Nir Barzilai, who's at Albert Einstein College of Medicine, is a scientist and a proponent of metformin for protecting against age-related diseases. So his opinion I hold very highly, but also I've looked into the data, and there are a couple of scientific papers that looked at over 10,000 people. There's one that's looked over 10,000 veterans who either did not take metformin or did. Okay, it's a prospective study, it's a retrospective study, so it's got some issues. But when you look at that data, it's really quite compelling, because the people who took metformin for their type 2 diabetes, right? High blood sugar is what you typically are given metformin for. Those patients, the risk of getting not just worse type 2 diabetes, but also if they were predisposed to cardiovascular disease or Alzheimer's or frailty, their risk went down. And those people who didn't take metformin, who, by the way, in most cases didn't have diabetes, their risk went up over time, over a matter of, I believe it was 4 or 5 years after that. That, to me, was really convincing that this drug could protect against those diseases. at least if you're high risk. We don't know about average risk people. But you mentioned risk and reward. This is important for me and for my father, who's a scientist as well, who has a lot higher risk right now at age 81. You ask yourself, what are the chances of it doing harm? And if it's almost zero, then you look at the cost of the medicine, and if it's cheap, the downsides are really low and the upsides are potentially high. The real risk, I mean, let's be honest, the real risk is aging itself. It's going to happen. There's no denying it. And for most of us, it's not pretty what happens. And so then when you balance those up, it's almost obvious to me that these risks are worth taking. But of course, people have different tolerance for risk and don't all have access to scientific papers and can judge them.
But to be sure, as you mentioned earlier, I mean, it was what I think our segue was, that exercise is also a powerful activator of the AMP kinase pathway.
Well, it is, and that's a relatively safe way to do it. It's not risk-free either, right? I've had a couple of friends die from infarctions recently that overstress their bodies, we think, to do that. But yeah, if you do moderate exercise and you do it over long periods of time, your risk of mortality goes down dramatically. There's one study where people who cycled a lot, and the number, David, I can't remember exactly, but it was quite a lot, let's say 40 miles a day, something like that. Their risk, overall mortality risk, went down by 40% in their later decades, which is a remarkable benefit. So yeah, I mean, if you're not interested in pharmaceuticals or supplements, exercise and fasting are two, I wouldn't say easy, but relatively available things you can do. But you want to start before you become frail, of course, because it's very hard to do this once you've lost your muscle mass.
Well, it takes us to another place, and that is the notion of the sweet spot, if you will. I hate to say that because it connotes sugar, but that said, between becoming frail and building up, I'm getting to this sort of dichotomy between the importance of autophagy, of breaking down, and the importance of building up, but not overdoing it. How do we get to that important place of determining what's best? In other words, how much breakdown of our tissues do we want to encourage versus how much buildup, as mediated by our stimulation of the mTOR pathway, do we want to encourage as well? So how do we look at that? What's your take on that? Because, I know that you are exercising, you are restricting your caloric intake, you're reducing your number of feedings. I hate to say that, as a laboratory animal per day, or how often you eat. I think you're probably paying good attention to your sleep. And so where does one find that balance? I mean, I know you know James Clement. He wrote a wonderful book called The Switch. We had him on the program. And it's really talking about that pivot point. Where do we determine that?
Well, there's no one size fits all. It's why in my book and in my life, I turn to devices like the one I'm wearing on my wrist. It's an Apple Watch, but there are plenty of others. I also have a ring, the Oura ring, O-U-R-A. And I also occasionally take blood tests and look at what's happening to my body, particularly if I change my lifestyle. I want to know what's happening. Otherwise, I'm flying blind and I don't know if I'm doing harm or good to my body. But I measure about 34 things. In full disclosure, that the company that I use, I'm an advisor to, but I want to mention it because I think your listeners will want to know what I do. It's called Inside Tracker, and it's a Boston-based company. And I've got 11 years of data on my body for these markers, and I can estimate my biological age relative to other people. But also I can see when things are starting to trend up with age or lifestyle, and I correct them as best I can. And so far, I've been able to keep my estimated biological age younger than I otherwise would be. Now, this is different than the Horvath clock, which I haven't measured yet, but I will.
Oh, I bet you're dying to do that.
Well, I'm a little bit worried that I'm going to get a bad result, but I want to do it.
You have to tell us, though.
Of course I will. I'm a scientist. I'll tell everybody. The other reason, David, that I'm a little bit hesitant is a colleague of mine took two tests from two different companies and got an 8-year difference in the answer. So I'm waiting to see where it falls and which one is proven more accurate, and then I'll do it, because I don't want a false reading and then either get
Well,
something.
I think it does kind of point to the accuracy of the various available now so-called Horvath, or at least DNA methylation tests that are out there. And, again, Dr. Horvath has been noted to indicate that his methylation measurements and that patterning, which there now are several. He has one called the Grim age, which kind of predicts when we're going to die. That was named after the Grim Reaper, but that it's been sort of inviolate, at least for those of us who are in shape. He did indicate that there is an impact of lifestyle changes in people who are overweight or have diabetes. There was one recent publication, I believe it was last year in October, looking at overweight African Americans. And the only intervention in those who had low vitamin D levels was to, at various levels, replete their vitamin D. I think it was 600 up to 2,000 IU a day, and demonstrated, in fact, changes in, I think it was the velocity or the rate of change of the Horvath clock, or it may have been the delta between chronologic versus age versus the Horvath age. But, really interesting information there that, in moving forward, I think it's going to be really terrific that we can apply that information to ourselves. I want to get back to knowing about yourself. You are the N of one, and I've been speaking to our viewers for quite some time about what I learned in the morning when I wake up and have on my Oura ring. And we did a nice interview with the founder of that company. What I learned about last night's sleep. Was it restorative? Did I get enough deep sleep? Did I have enough time in REM? Did I have enough total sleep? And then tweaking modifications for me as the N of one. I wear a continuous glucose monitor. We recognize how you've talked about today, how important knowing your blood sugar is, and how the choices that I make in terms of my diet, my periodicity of eating, my how I honor my chronobiology in terms of when I eat, and how that affects my blood sugar. And I am, full disclosure, an advisor to a company called Levels Health that, while they don't provide the continuous glucose monitor, they then deal with the data and give you good information about changes that you can make. And then, laboratory studies that are really generally available. I mean, I think it's really valuable to know markers of inflammation, your hemoglobin A1C, and your uric acid level. I think in moving forward, we're going to hear a lot more about the relationship between elevation of uric acid and many of these downstream effects that we talk about in terms of inflammation and metabolic disease. But, as we close, we've covered a lot. And I would like to just ask you, as you're falling asleep at night, imagining where the future takes us in terms of where you are right now, especially as it relates to this incredible study that you just published. And I will say, the press took your study and said, laboratory reverses vision loss in laboratory animals. Really, I think perhaps missing the point that you revealed, that mammals retain the information about being young and having vitality again, that it's all still there. And that, your metaphor of it's there on the DVD, but it is just scratched over time, and that you're able to buff the DVD and allow us to listen to this incredible music of youth and vitality again, that it's there within each and every one of our cells. And that, to me, represents the incredible, empowerment of where this may go. So let's say we play it forward in an ideal world, that you have this keep this incredible laboratory, unlimited funding. Where could we be, let's say, 10 years from now?
Yeah, well, so I was hoping in my career that we would help our children and our grandchildren, but things have gone so quickly. It's really a pleasure to be alive right now, and honored to be part of this. I think about it a lot, David, of course. What I did as soon as we made this discovery was to think about how can we apply this. And I've spent some of my career doing clinical trials, starting biotech companies. Because we treated the eye we could immediately imagine treating glaucoma. And so we're working towards that. So in the near term, I'm hoping that we'll be able to use the AAV, the adeno-associated virus technology, to, for the first time, restore vision in patients with glaucoma. Right now, there's nothing that you can do to reverse the disease. Very poor prognosis. Perhaps macular degeneration, perhaps damage to the optic nerve, maybe spinal injury could be reversed. But you're right. I'm glad you picked up on that, because it was surprising that the media didn't really grasp what I think is the main discovery here, which is that all cells in the body seem to have this ability to reset, not to zero, because that would just be fairly easy, but getting them back to being young, but not so young that they're dysfunctional. How do cells know how to do that? Where is that information to be young again? Where is that stored? That's still a mystery. But I can tell you there are a lot of scientists now who are searching for it. And when we find it, it's going to be a big deal. I hope that within our lifetime, we see this explode, and that we'll be able to treat many different diseases of aging. You could reverse the age of your heart. You're heading for heart failure. You turn the age of the heart back to 30 or 20. Ultimately, where this is going, I think maybe in our lifetimes, maybe for our children, is to reset the age of the body, whether it's with gene therapy or perhaps one day just a pill that you take for a few weeks. You wind the clock back, and your body isn't just acting young, it literally would be young. And if you have a young body, you don't get heart disease, you don't get Alzheimer's. There was a recent study by my colleague Manuel Serrano who reprogrammed the brain of old mice and got neurogenesis and memory back. These are things that I hope we'll be able to achieve. Now, just in case it's not clear, we have to be very careful, because if you go too far and too quickly, you can cause problems, you can cause cancer even. But we were very lucky with our treatment. These 3 genes, even though we pumped the mouse full of them, either just in the eye or the whole mouse, there was no sign over a year of any increased cancer. So we think we've just lucked out and found what is this ancient reprogramming mechanism that probably helps one of these salamanders regrow a limb, or helps our liver regrow the rest of the liver if you cut a piece out, and regrow the optic nerve if you do what we did.
So the refinement then of the factors was you went with 3 of the 4, and the other nuances would be that have already been explored in laboratory animals has been in adult animals, how long they receive it, and turning it on and turning it off. And you do have that ability then to turn it on and turn it off.
We do. So we engineered the virus. We've got our own special brand that if you give doxycycline, the antibiotic, the virus is engineered to respond to the drug, to turn on the genes. And then when you take it away from the mice, they just drink the water with doxycycline, it switches off. And the same can be done for the patients that we hope to treat. So you could imagine that the doctor gives the injection in the eye to the glaucoma patient, take these antibiotics for 3 weeks, come back, we'll measure your eyesight. And then if it's restored enough, you stop taking the antibiotic. But what gets interesting is if your eyesight ages again and you lose your eyesight, you just take another course of antibiotics and you just repeat that process as many times.
To activate that pathway.
Right. We don't know how many times you can reset the system. It could be once, it could be 50 times. I would say that it's going to be more than once for sure.
Well, as we close, I wanted to tell you that at the end of your book was it's very sweet what you did, and I think when our viewers read your book, they're going to feel the way I do about it, and that is, you basically talked about what it's like to walk around your laboratory, and you honored every person who works there, every person who's contributing a small but obviously very important piece of this grand puzzle. And I despite all the wonderful information in your book, that's the part that really got me. I mean, you're a gentleman, and it was very meaningful to see that. So thanks for doing that.
Well, thanks for saying that. They're my second family, and they work really, really hard, and it's been particularly hard during the last year for them as well. But there are tens of thousands of young scientists who really don't get the credit they deserve in labs really all around the world. You know this, David. And they're striving to be the 1 in 100 people who gets a successful lab. And it's a tough career. And of course, it makes sense that they are the ones that should be the heroes in these stories.
Well, in closing, you have made yourself somewhat available, at least in terms of your work. There's a website that shows delineates your research. So where can people go to learn more about what you do, obviously, aside from reading your book?
Well, I have a website for the book. It's called lifespanbook.com. And I have a number of newsletters you can subscribe to. And that's the main one. I'm on social media, on Twitter at David A. Sinclair, and Instagram, David Sinclair, PhD. And yeah, I try to put out a variety of things, highlight new findings that are relevant, and give my 2 cents about how we should fight for science and the truth. But yeah, you can find me there. And that's a good place. I mean, other than that, I'm writing another book. And so stay tuned for that one as well.
Well, I'm going to end on a quote from Ralph Waldo Emerson. And if I have used this previously in a podcast, I don't recall, but if I did, I'm going to use it again. And it says, "Do not go where the path may lead. Go instead where there is no path and leave a trail." And that's all about you. I mean, you're breaking new ground, and people are following you, and you're welcoming them along. And with that, let me just say thank you for spending your time with us today.
Well, David, thank you for this platform, the people that you bring on. It's just a gift to all of us who can listen directly to the scientists and these pioneers, which even just 5 years ago we didn't have access to. So thank you as well.
Okay. All the best. We'll talk soon.
Sounds good. Thanks.
Wow, that was an incredible program, wasn't it? We just had the opportunity to explore the leading edge in the science of longevity. And it really looks like there are going to be some incredible developments that are going to be translatable to us as humans in the next 5, 10, perhaps 15 years. But in the meanwhile, what else did we learn from Dr. David Sinclair? In his book, Lifespan, Why We Age and Why We Don't Have to, again, here's the book, we learn some powerful tools that we can implement right away. Things that we've talked about before in the program, like eating less often. Don't know who developed the 3 meals a day idea, but as Dr. Sinclair made very clear, that might be something we want to avoid. Eating less calories. Both human studies as well as certainly the rodent studies have demonstrated for quite some time that reducing our calories is a good thing, that we stimulate pathways for longevity when we reduce our calories. Also the notion of intermittent fasting. Many of you might have participated in our fasting online program. We've talked a lot about intermittent fasting on the program before. Dr. Sinclair advocates eating less red meat. We know that red meat, things like beef, and as well lamb and eggs, have certain types of amino acids that will go ahead and stimulate the mTOR pathway, which is something that we should probably cycle through, that there are times when we should reduce stimulation of this mTOR pathway that we talked about today. Having said that, having red meat from time to time might well be okay, but we want to think about the idea of cycling away from constantly stimulating this mTOR pathway. We talked quite a bit about exercise. What a powerful way to increase NAD, which was certainly a big topic in our discussion today. Can't say enough about the importance of exercise through stimulating of what we call the AMP kinase pathway. And finally, how interesting it was that we explored the stress upon the body that it is induced by exposing ourselves to both heat and cold. The idea that these are challenges that further increase our production of this wonderful chemical called NAD that then goes ahead and activates the sirtuins, which we really want to do. And so much groundbreaking work in that research, again, by our guest today, Dr. David Sinclair. So finally, here is the book once again, Lifespan, Why We Age and Why We Don't Have to, Dr. David Sinclair. That's all we have time for today. Thank you again For joining me, I'm Dr. David Perlmutter, and we will see you soon. Bye-bye.